This is the third in a series of presentations on seasonal affective disorder, our biological clocks. We know seasonal affective disorder has a lot to do with daylight and darkness. But more than that, we've learned, really, that it's all about synchrony. Circadian clocks govern human biological processes and behavior. They are our timekeepers. We're most familiar with external or environmental time, the circadian rhythm of daylight and darkness, and the changing ratio of daylight to darkness through the seasons. All living organisms also have internal circadian clocks. Humans have a master clock or timekeeper called the suprachiasmatic nucleus, or SCN. This tiny control center within the brain is responsible for all our circadian rhythms, including sleep-wake cycles, changes in body temperature, blood pressure, and heart rate, and release of many hormones. The suprachiasmatic nucleus is located in the brain's hypothalamus. The eyes are the pathway to this master clock. Embedded in the inner retina are spidery cells that signal the SCN. In turn, the SCN tells the body when it's time to go to sleep, when to be hungry, when to wake up. Let's take a closer look at the suprachiasmatic nucleus in a three-dimensional animation from a 2000 lecture by Dr. Joseph S. Takahashi, a specialist in circadian biology.
So what I'd like to do in the second half of this lecture is to really tell you about what we know about the control of rhythms in mammals. And in humans, we synchronize to light, and that light is received by our eyes and the retina. And this information travels down the optic nerve into the base of the brain in a region called the hypothalamus. And within the hypothalamus sit two very small wing-like structures shown here in yellow that are composed of a few thousand neurons. And we know now that these structures actually contain our biological clock. The suprachiasmatic nucleus, or the SCN as we call it, acts as a biological clock system for us in our brain. Now, if we look inside the SCN, we find that it's really made up of a network of nerve cells, many thousands of them. And interestingly, these nerve cells fire with a circadian rhythm, and I'm going to show you an example of that at the end of my lecture. This kind of experiment has led to the idea that the clocks in the suprachiasmatic nucleus are actually cellular clocks. And so it is within the individual cell that the clock mechanism really emerges fundamentally.
So humans and almost all organisms display internally driven circadian rhythms. The basic properties of the rhythms are all the same. They keep us on a 24-hour schedule, approximately. They synchronize to environmental cues, predominantly light. And significantly, they are maintained even in the absence of such cues. That is, even without the cycling of daylight and darkness. But what happens without these external cues? How are the natural rhythms of human life affected by living, in a sense, beyond time? The answer to these questions was, in fact, a goal of temporal isolation experiments dating back to the early part of the 20th century. The earliest experiments were conducted in caves or bunker-like apartments. Among the most well-known were experiments conducted by Michel Seyfried, a French speleologist, in the 1960s, 1970s, and again in 2000. The objective of these studies was to determine if people in complete isolation would continue to fall asleep at their usual time, or if their sleep-wake cycle would begin to run too fast or too slow. Among the findings, Researchers discovered first that a subject's circadian rhythms persisted despite the isolation, proof of an internal master clock. But these experiments also showed that this clock was not perfectly accurate. It lost a few minutes every day. That is, the human internal circadian cycle was slightly longer than 24 hours, roughly 24.5 hours. This may not seem like much, but if someone's cycle lasts 24.5 hours instead of exactly 24 hours, then everything that a subject used to do in the daytime eventually would end up being done at night, and vice versa. Depending on the length of isolation, the cumulative effect could be a miscalculation of the actual date at the time of emergence from isolation, by days, weeks, or even months. Isolated from environmental cues, we follow free-running circadian rhythms. In a sense, we're drifting in time. For example, this chart shows for the first 9 days of an experiment, the subject was exposed to the natural variations in ambient light and noises that characterize day and night. For the next 25 days, the subject was cut off from all such cues as to the time of day and was left to operate according to his own internal rhythm. He continued to display a sleep-wake cycle, but it lengthened to about 25 hours. In the last 11 days shown in the diagram, the subject was placed back in normal alternating conditions of daylight and darkness. The length of his cycle returned to normal. Even more surprising than the subject's desynchronization was the finding that once the experiment ended, it took only a few days for him to resynchronize his cycle to external daylight and darkness time cues. We know that with jet lag and shift work, we always readjust to environmental time. Temporal isolation experiments have shown us that what is really happening is that we are resetting our biological clock. Seasonal affective disorder also involves desynchronization and resynchronization of our biological clock. And we now know how to help along that process with bright light therapy. Thanks for listening to this presentation. Other presentations can be accessed from this webpage, including treatments for winter SAD with a focus on light therapy, seasonal affective disorder, it's not just the winter blues, and winter SAD and the environment.